I didn't even hear of chronobiology until approximately 1976 or thereabouts. I came from a background in neuroscience, neuropharmacology, and was fortunate in neurochemistry was my perspective. So I was fortunate in having the opportunity to go to Axelrod's lab at NIH, which aborted my residency in psychiatry. And I seriously considered that a contribution like his to psychiatry would be about as good a way of being a psychiatrist as there could be. And so that was my interest. And that's where I was introduced to the pineal as a model system, a very fine model system, and was spoiled immediately by 100-fold effects, which were very easy to investigate, rapid, fast, specific assays, and all other sorts of things that spoiled me for the difficulties to come. So occasionally circadian features, which were seen as tools or phenomena, came up, but it wasn't the goal of the research. The goal of the research was transduction mechanisms and neurotransmission and its consequences. Pineal was already a good model system for that. The circadian aspect was another set of tools and another set of dynamics. So I was interested in the phenomenon of regulation of sensitivity. That is, that if you give an injection of isoproterenol, an adrenergic agonist, the norepinephrine analog in some respects, you get a very strong response in the pineal. And then if you wait for that response to go away and do it again, you get a much smaller response. In contrast, if you block such responses, either by propranolol or by denervation at the level of the ganglion, the response you get is much higher. Well, that is a history-dependent effect, which in its essence seemed to be important for psychiatry.
Why?
Because throughout the 20th century, it was felt that psychiatric problems were often a result of previous experience, especially in childhood, with residual effects. And the physiology of residual effects, if you will, was not understood. And in addition, connecting more to the circadian aspect, the circadian system conceptually is a beautiful model for the interaction between endogenous and exogenous stimuli. The response not only depends on what the stimulus is and its history, but the state of the system at the time of the stimulus. Again, this is the sort of complexity, a simplified version of the sort of complexity of people's responses to the same stimulus varies as a function of their own personal history, the current state, and their experience with whatever the stimulus is. So this was the abstractified, motivating, psychiatric perspective, which I mostly since have given up. At one point, Mike Menaker's name came up, and he'll play a role in the early days. And I remember well asking Julie about him, and he said, "Oh yeah, he's good, but he does this woodsy fieldsy stuff." And sure enough, I ended up being far more woodsy fieldsy than I had ever thought in terms of things to read and attend to. Just accepted papers on strange critters. So in fact, I went to a meeting in Jerusalem in about 77 or thereabouts, and met Mike Menaker, and had the opportunity to ask him about rhythms, and especially asking him about some papers that I had recently read by Pettentrig, which did not particularly make me attracted to entering the field. They were highly theoretical, highly abstractified model papers, and about maggots, eclosion, and flies. And it was a theoretical attention to such things. I was not particularly interested in either the theory or the system, and asked him whether I had to understand these papers fully. And it was a watershed reply, because he said no. Had he said yes, I would have been gone. So the next critical step in your question, which was how did I get into this business, is that there was a meeting in Japan sponsored by, I think it's called the Naito Foundation or something like that Julie was invited to. And he passed it to me. Well, a trip to Japan, one of the nicest meetings I've ever been to. Got a nice watch, which we would show each other as a badge, all the people who had attended that meeting. But it was far enough in advance so that I was wondering how to either gear my work into a talk that would be relevant, or maybe do some additional work that might be relevant. It was about a year and a half or so in advance that I knew of this. So I read some more literature. In fact, it might have even been before this Jerusalem meeting that I learned of the Japanese meeting. So I read a number of I read The Burning Book. There were these big fat neuroscience compendia books. I've forgotten what they're called by an outfit that exists. The Pettentrig had big articles in. There was a series of German meetings that that were then published. The proceedings were published, and this was a very fine one. In those days, they were very interested in solidifying the existence of the clock in the sense of its being autonomous and independent of all other things that might mislead you into thinking about other causes of the phenomena being studied. So the homeostasis of tau was very important in those days, and the imperturbability of the clock by anything except the physiologically evolved mechanisms of particularly light was something that was important to establish. So I saw these sentences that said that the clock was impervious to pharmacological intervention. Now them's fighting words for someone like me, because after all, we already knew that the pacemaker was in the SCN, and that there was a synapse from the retina in a special pathway to the SCN, which means there must be a transmitter. And if there is a transmitter, then there is a receptor. And if there is a receptor that has anything to do with anything we know about, then there will be an agonist and perhaps an antagonist. Might not apply to unknown peptides, but generally cells will have multiple receptors and multiple responses to not necessarily the transmitter, but other neurotransmitters, agonists, antagonists. And so it cannot be true. And so I simply started injecting neurotransmitters, agonists, and the usual ones, serotonin, norepinephrine, and such, to see what they would do to the rat pineal rhythm. And the one that worked quite well was carbachol. And the fundamental meaning of this for me was the clock is susceptible to pharmacological perturbation, which means it's susceptible to pharmacological investigation. So this paper was published, and I got invited to a Gordon conference, and I witnessed the, at least for the Gordon conference, the final battle of the Pittenregians allied with the Ashafians taking over from the Halberagians. I felt that the right side had won, and there I was. So I became a chronobiologist. Well, let me add another element of the transition for me. As I said, I started with an interest in history-dependent effects and causes of variation in response, and sensitization, desensitization is a very fine phenomenon. But the PRC took that one step further into the mystical realm. Not only was the size of the response a function of the history, as in desensitization, but the direction of the response was a function of the history. Now, that may be because the term is measured in a different way. Nonetheless, you can look at the graph. It goes up, or it goes down, or it goes nowhere. Now, that is a history-dependent response, and that's what really made me interested, in addition to the question of whether neuropharmacology could be used as an approach.
What you were talking about in your lecture was about Axel Rod's way of doing science. Can you talk about that? You know, you said you had one question and you did one experiment and you got one answer. I mean, the way he did research and how it influenced you, obviously.
His style did not appear to be all that sophisticated or deep. Questions, once you're deep into it, seemed rather simple, obvious, and straightforward. So the goal of an experiment or a project did not require paragraphs of explanation. They could be stated in a sentence or two and understood if you knew what the words meant and if you knew something about the literature. Retrospectively, that's an amazing ability. Here we are at the SRBR meeting, and look how much you have to understand just to understand what the question or the experimental goal was, and how complex the experiments are to address these questions. So that was one of his traits. Another was he developed assays, and he particularly used S-adenosine methionine and methyl transfer as a way of finding an enzyme and then partially purifying it. And then he would use that enzyme and the methylation as a way of measuring the transmitter. It was an incredibly rich approach, and experiments would be fast. The assays would be convenient, often extraction assays. They would be specific, they would be fast, and they would be quantitative. So you could run through a series of developments, questions, and advances in a remarkably short time. It never occurred to me that it in turn made it harder to keep on going, because you answered the critical questions quickly. Now what do you do? What he did was he shifted to other questions. And so there was a talent of rapid progress, and then letting it go when it got too hard. Sometimes he would come back to it 5 years later when something came up that allowed progress to be made quickly again. So the ability to get in to a new question, which wasn't grossly new, but there was a new opportunity of some kind that he could make, and then shift was an amazing talent. Very few people um, could mimic it. Perhaps Sal Snyder came as close as anyone to having a similar style and being as successful. So
You,
persistence, headbanging, was not a trait. Other people, there are many styles that work in science, and there are certainly major advances and major figures whose trait of persisting on an important problem led to their success for an important problem. What he did, as he himself said, was he followed his nose. One step at a time. Each one led to the next. And when it got to be too complex or too difficult, he would go somewhere else. And he did later add, just follow your nose, but you have to have the nose. Well, I first met Bill Schwartz was at the NIH at the same time I was, but I met him at that Japan meeting, at that Naito Foundation meeting in 1978 or thereabouts. But I met him there for the first time. His personality is much as it was all along. And one thing that happened at that meeting in Japan, which I treasure and which illustrates him, is he bought a robot toy, a little robot car with a radio transmitter that controlled it and made it go. This was new at that time and unique to Japan. And we were treated very well by the hosts. It was a relatively small meeting. I don't remember whether it was 30 people or something like that. There are pictures of it that are fun to see because a lot of figures were there. So he bought this toy for his, and I don't think he was even married at the time, but he bought this toy, which was intriguing. And we were being put up at this fancy Tokyo hotel across the street from the Imperial Palace. And for reasons that I don't remember what triggered it, he opened the box and started playing with this toy in the lobby of this very fancy hotel on a marble floor. And so this toy was zipping around and he was standing there with his little controller having a grand old time. Everyone was in suits. And this circle of Japanese businessmen formed around and watched this little car going around. And their serious mien broke into smiles and wonder. I think at the toy and at this, I don't know what the word, barbarian, who was playing with a toy in the lobby, which occupied space as well. They have to go around if they wanted to ignore it, in the middle of the Tokyo hotel. But it was so delightful that they simply stopped to watch. And I think it's a lovely story because it's not a prank. That is, it was just sheer playfulness.
Delight.
Yes. Which was radiating, which was catching.
That's a nice. Let's go back to NIH as it's changed over the years. Because when I went there, it was a sort of a high. And you had an idea with X, who you met in the corridor on Tuesday. You did it on Thursday.
That was literally true. And many
That's
people... Many people's memories, going back even further, but it was still true at the time that I went there. There was a sense that today is Thursday. On Monday I can do anything I like, including an entirely new project, or a different discipline, or a different sort of question. And that wasn't absolutely true, of course, but it was true enough and happened enough so that Julie Axelrod's story about meeting I'm sorry, the name escapes me at the moment. Someone standing next to him at the urinal asking how to measure enzymes and being told, well, you just take a rat liver, cut it up, maybe grind it up, and put and so on, and then doing it was literally true. You could. It doesn't have. You don't really have to go to the urinal. Um, there were no.
I remember
and,
the lift. You know, you're just waiting for the lift.
Yes, the hallways
The
basically.
hallways.
Actually, one of the dictums, one of the maxims you can come out of is it's important to have crowded labs. You don't need administration to foster communication. You need crowding. So many collaborations and especially many consultations was the norm. Every time you walked down the hall, you had the opportunity to consult with someone, or that person would consult with you, so that you knew what was going on down the entire hallway, which might have 20 investigators, and that they were available to you for consultation or collaboration. And these things you could turn on a dime if it didn't cost too much, if it didn't take too long, but there was nothing that determined. It felt like there was nothing that restricted you from doing anything you liked that would take a week and not cost very much.
It was a very special time.
Furthermore, there was the sense of opportunity rather than planning. You kept your ears, you kept alert, you read the literature, you listened to what people had to say, and you had the opportunity to jump to a new question or a new system or whatever, rather than fulfilling some plan that you had submitted at some point that had gone through a system of approval. It's not that there wasn't any, but the general atmosphere was an atmosphere of scientific freedom and of bottom-up initiative. I was unconscious of an administrative structure above the lab chief. Julie Axelrod was the section chief that I worked in, and Irv Copen was the lab chief. Irv Copen was there, available, and doing active research. And the reality of the hierarchy was not palpable. It certainly was not, it was a very collegial, intense system where, for example, there was it wasn't really very different meeting Irv Copen in the hall and his asking, so what's new? And meeting a fellow postdoc who happened to work with Irv Copen, who asked what's new. So I was certainly unconscious as a postdoc of directors, assistant or associate directors, NIMH directors or NIH directors, and plans and guidance, which became more and more structured, just wasn't there, at least not palpably.
And it changed slowly, or?
It's very difficult to know how much. Well, this is a classic problem. It's hard to know how much distinguish between how much times have changed, and how much your experience has changed what you see, and how much what you're exposed to or now can see that you didn't see before, which was always there, changes. And it varied, and it varies with individuals. For myself, the freedom to work that I felt I had persisted for a long time, and I didn't. And I worked with my own hands for a much longer time than I realize is the norm. I didn't know that was the case. I didn't know that I was supposed to leverage each success into an entrepreneurial venture of accumulated resources and people. And so that kept me under the radar in many respects for a long time. One image that I felt was that I stayed in my cave and it wasn't particularly noticed. The opening wasn't particularly noticed by the higher-ups until one day I got a little jealous of some peers and moved the rock from the front of my cave. That was a mistake. Because then they knew where the cave was and what was in there, and I could not go back and hide again.
If you go back to your pineal years, pipetting in your cave, what are the highlights and what were the best bits of trying to dissect out the pineal function in your model?
Well, I left the pineal after the initial forays into the circadian aspects for rat pineal, back toward brain neurochemistry, and worked on acylation of protein. And for a while there was some lipid work. Actually, I remember a project having to do with lipid methylation and finding some unknown spots. And one of these presented a really classic dilemma. You see, if you're looking to identify an unknown, which you happen upon, you have no way of knowing how important it is until you find out what it is. And you don't know whether it's worth trying to find out what it is until after you found out what it is. So you have no way of knowing when to get out. And the answer is, don't start unless you have some reason to start. But the temptation of, well, it's unknown, it might be important, can suck you in. So I spent a few years on that, and also spent a few years on protein acylation, which turns out to be quite important and interesting. And then went back. And this time when I went back, I went to back to the chick pineal, because the rat pineal is photosensitive and rhythmic in vivo, but not in vitro. The chick pineal has it all in vitro, which is a much more convenient system. In the interim, Joe Takahashi came and worked in my lab. He was supposed to be doing this, but he did science on the phone a lot. And when he left the lab, he did. But he was a very highly developed scientist. When I met him as a grad student, I think he sprang fully formed from the head of Mike Menaker.
Why did he come to the postdoc with you?
His background was biology and circadian rhythms, and I think he wanted to learn more biochemistry and subcellular approaches. Is the simple answer. But the intensity of his desire to work at the bench was lower because he already knew how to organize work without necessarily being the one to do it, which I don't know that I ever learned properly. So there was some issue of my urging him to do experiments. In fact, he was being productive on the phone. I didn't know that you could do that. And again, it's a matter of a deeper understanding of the system. So for example, shortly after he left, he started his own lab and he was working, and I said, how are you doing? What are you up to? And he said, well, we've become interested in such and such. I could not understand the word we. Who's we? I didn't ask about we. I asked you. And that sense of organization was quite different. And of course, I wish I could have done it his way.
I'm going back to you.
But back to me, there was something about, there is something about the sense of autonomy as a scientist. And that for me was connected to doing it with my hands, because then I knew what was happening. And I always had trouble understanding how to figure out what went wrong, because things are always going wrong. Uh, if I wasn't doing it, I don't know how the candidate problems can be so trivial that it's insulting to ask. Did you plug it in? Did you check the setting? Um, was the centrifuge at a different speed because of the person who used it before you? These are not fair questions to ask, and yet those are the kinds of things that go wrong. So the sense of autonomy was tied to the sense of autonomy of what I'm doing at the bench, more than the group. But that should be a stage of development.
And your work subsequently on chick pineal.
I still think that chick pineal cells are a great system, because aside from having an entire system in a dish, which is still not so common, that is, they are photosensitive, they are rhythmic, they are responsive, and so on. Current systems are not always photosensitive at the same time as being rhythmic. They may be one or the other. But one of the things that was important for me is, because I had been working in the rat pineal, acute, let's call them acute effects, which in circadian jargon is also called masking, and which I consider highly physiologically relevant, although the circadian tradition is to try to exclude them rather than include them. The chick pineal showed acute effects simultaneously with clock effects, which provides an internal control, which I found extremely valuable. It allows for dissection of pathways, because we could track the acute pathway is somewhat similar, reasonably similar to the rat pathway, cyclic AMP and so on, and we could track that and test those elements against clock effects, and they would commonly work on the acute effects, so everything's fine, but not work on the clock effects. So I had a negative conclusion with positive results. That is, positive for the acute, negative for the clock, which I could have confidence in. And that was a great strength of the chick pineal cells, although I always found it embarrassing to say I worked on chicken, because people would ask me questions about chickens. When I got a reprint request from the Department of Poultry Science, I cringed. How have I come to this? I was supposed to be a neuroscientist, neurochemist, biological psychiatrist, whatever you like, and I'm working on chickens.